Don't hurry. You're right. Hello, everyone. Now, can people up the back hear me okay? Terrific. Thanks for the thumbs up. So welcome, everyone, to tonight's Menzies Institute for Medical Research Roadshow. For those of you who don't know me, my name's Tracy Dixon. I'm the director at the Menzies. Before we start today's program, I would like to acknowledge that today we're meeting on Lutruwita, Tasmania, and that these are Aboriginal land, sea and waterways. I acknowledge with deep respect the traditional owners of this land and the ongoing custodians, the Palawa-Pakana peoples. Our island home is deeply unique, with spectacular landscapes and with our cities and towns surrounded by bushland, wilderness, mountain ranges and beaches. And I acknowledge that it's a privilege to stand on country and walk in the footsteps of those before us. Yesterday, I actually had the privilege of being on country with some of my university colleagues, and we took time out in a pretty hectic couple of weeks and months to take a breath, have a chat together, listen to the birds and feel the sun. And I felt myself actually physically re-energised. And it reminded me that we often rightfully reflect on Indigenous people caring for country, but we must also remember that country, that nature, that the places we all care about across Tasmania, they take care of us too. So I stand for a future that profoundly respects and acknowledges Aboriginal perspectives, culture, language and history. So it's a great pleasure to see so many of you here tonight, especially on such a drizzly night. I really appreciate you coming. So welcome to our valued Menzies donors and supporters, Professors Rufus Black, Natalie Brown and Graham Zosky from the University Leadership Team, Councillor Richard Ireland from the West Hamer Council, our friends from Clifford Craig who are with us, our colleagues and collaborators from the Tasmanian Health Service, our UTAS colleagues from Launceston and those that have travelled north for the graduation ceremonies. Welcome especially to the people who are new to Menzies and are hearing about our work for the first time. So I'd also like to acknowledge the many people in the room who have lived experiences of the conditions that we'll be talking about tonight. Welcome to everyone. At Menzies, our mission is pretty simple. It's to perform internationally significant medical research that leads to healthier, longer and better lives for all Tasmanians. We have over 180 research staff and we're building the medical research workforce of the future with more than 100 PhD students. We work across key areas that affect our community, including cancer and genomics, prevention, health services and wellbeing, environmental and respiratory health, cardiovascular health and brain health. Our work is truly bench to bedside, from innovation to translation, with some of our researchers specialising in public health and epidemiology, others in clinical research and others like myself in the laboratory. Although our buildings are physically located in Hobart, our work is explicitly designed to have impact across the entire state, as well as nationally and internationally. So tonight you're going to hear from three of our researchers whose work holds so very true to our mission of performing internationally significant research that has local impact. The evening will be structured so that you'll hear from each of their presentations and then I'll invite them back to the floor to take your questions. So with no further ado, I'd like to introduce our first speaker for tonight, Professor Catherine Burden. Catherine is a geneticist and leads our iGenetics group. Her research is dedicated to using genomics to uncover the causes of childhood cataract and glaucoma. Catherine is also the Deputy Director at Menzies, providing leadership across our terrific cohort of PhD students. I'll hand over to you, Kath.
Okay, thank you. Is that microphone working? Yep, great. Thanks, Tracy, for that introduction. As Tracy said, I am a human genetics researcher, and that means that I spend most of my research time trying to understand the changes in our genes or our DNA that affect our health and lead to disease. I've spent most of my career working in genetics of eye disease, and I'm going to talk tonight about one of my pet projects that I've been working on for probably over 25 years now, and that is childhood cataract. So what is a childhood cataract? It's the same as an adult cataract, but it happens in childhood. It's the opacity of the lens of the eye. So that in the top right there is a diagram of the eye as if you're looking side on, cross-section through the eye. And at the very front, you've got the clear cornea, which protects the eye. And then you've got the coloured disc, the iris, which is, if you look front on, is the blue or the brown part with the black pupil in the middle. And directly behind that sits the lens. The lens is a really specific structure. It's got exactly the right 3D shape and refractive error that it needs to focus the light onto the retina at the back of the eye. So if that becomes opaque, the light can't pass through it, it can't focus properly, and we get a very blurred or glary image. So childhood cataract is very rare compared to adult cataract, only affects about 2 per 10,000 children, but it is difficult to treat. A lot of the children will have a permanent lazy eye. There are a lot of complications from the surgery when you're doing these operations on very young, still growing eyes. And of course, the child has this for their entire life. So the impacts of a cataract during childhood can be quite severe and lifelong. There are a few different causes. Some of them include things like toxin or medication exposure. Trauma to the eye or the head can trigger a cataract. And historically, the most common cause was maternal rubella infection while the child was in utero. That has almost completely been eliminated with our vaccination programs for rubella. So that's a fantastic public health outcome over the last 50 years or so. But in other parts of the world, it's not like that. So we are very privileged to have that option here. Once we take rubella out of the game, we're left with genetics as being one of the most common causes. So this is where a change in the DNA of the child themselves has disrupted the biology of the lens and it causes a cataract. So we know about 80 genes that cause an isolated cataract, which is where the child only has a cataract, and a couple of hundred more where there are other features as well, things like intellectual disability or hearing loss, a range of other developmental disorders can come along with the cataract. Often in those situations, the cataract is the first detectable sign of that syndrome, and it can be a great way to catch the diagnosis early if we're on top of it and we do the genetic testing. So genetics is a useful tool in childhood cataract for providing an accurate diagnosis or refining the diagnosis so we know exactly what kind of cataract the child has and what that might mean for their future. So if we know the cause, if we know what kind of cataract they've got, we can understand what's going to happen to that child, determine their prognosis. And depending what kind it is, there may be prevention or early interventions we could potentially do. It might give us some clues as to what surgical complications they might experience. And there's quite a large literature that shows that actually just defining what the diagnosis is and why it occurred brings a lot of well-being to the parents. That just understanding what has gone wrong, that it wasn't anything they did, that they couldn't have prevented this is actually very calming to a lot of people and is a really good reason for actually going forward with the diagnosis. On the other side of the picture there are some of the things that families find useful about knowing the diagnosis. We can use it to predict the risk to future children in that family. So once we know which gene it is, we can make some calculations about the chance that a future child will have that disease, and I'll show you an example of that in a minute. And then families will use this when they're planning their families, what they might do in the future. And knowing the diagnosis actually opens up access to things like prenatal testing or pre-implantation genetic diagnosis, which is accessed by some families with the more severe developmental syndromes. So the GenCat project has had a variety of names over the last 25 years, but current iteration is called GenCat for Genetics of Cataract. The aim of this program of work is to identify the genes that cause childhood cataract across all its forms, and then use that knowledge to enable comprehensive genetic tests that will inform the clinical management of the children and their relatives. So we have recruited families from across Tasmania, Victoria and South Australia, and we have a few from Queensland and WA as well. So we're expanding nationally. And we collect information about each member of that family, if they have cataract, if they've got other things that go with that. And we collect saliva or blood for the genetic analysis. And this is the current team on the right there, a range of ophthalmologists, orthoptists, genetic counsellors, and of course the odd molecular geneticist thrown in for good measure. So I've said gene and genetics a lot. What does that mean? A gene is a piece of DNA that codes for, or that provides instructions for the cell to make a functional molecule, which is usually a protein, or sometimes it's called an RNA. But the key point is that the code tells the cell what to do and how to do it. DNA itself is made up of 4 letters. We call them bases or nucleotides, but they're just letters. In alphabetical order, they are A, C, G, and T. They're arranged in a linear string from left to right, and if you printed it out, it would look like that picture of the book there. The important thing is that the alteration of just one of those letters can cause a disease, including cataract. And if you printed out the human genome in its letters like that, it would fill 130 encyclopedia-sized books, fill a whole bookcase. And if you read it out at 1 letter per second, it would, for 24 hours a day, it would take you 100 years to read it out. So it's a big place, the genome, and we have our work cut out for us to find those one changes that cause these diseases. So this is an example of one of the families in the GenCat project. This is a pedigree diagram where circles represent females and squares represent males. And so here you've got, and when the cell is coloured in or black, that means that person has the cataract. So this is a diagram of a young child diagnosed with cataract at 2 years of age and her parents. So this child came into the project and we sequenced her genome and looked at all the genes, all 280 odd genes that we know can cause a cataract, and compared that to the sequence of those genes in her parents. And this is what we honed in on. This is one way we can represent the DNA sequence as it comes off the instruments in the laboratory. Each coloured peak represents one of the different letters. So I've written the letters at the top, but you can see, hopefully, that the 2 parents have identical sequence at this location. And if you look where that red star is in the middle, they both have a blue peak, which means a C. Now, looking at the daughter underneath with the cataract, at that same location where the red star is, she's got 2 colours, a blue one and a red one. Now, the blue one is the one we expect. It's the one both her parents have. It's what we think of as, we call it the wild type or the normal non-disease causing version of the gene. But somewhere along the line, one of her copies of the gene, because you have 2 copies of each gene, has turned red at that location. So the C has changed to a T. That is enough to cause Nance-Horan syndrome. It's a change in the NHS gene, which is one of the first genes that the GenCat project identified back in the early 2000s. And it comes up a lot in our genetic analysis over the last 2 decades or so. Now, it's interesting, it's really interesting actually, that neither parent has this change. So this variant has arisen in the child themselves, either in the very early embryo, just after conception, or maybe in the sperm or the egg just prior. We can't tell from this data, it doesn't actually matter. But the important thing is that because neither of the parents have this change, their future children are very unlikely to be affected by this syndrome. So that's very helpful to know, quite reassuring. But on the other side of the scale, for this girl, her future male children have a 50% chance of having Nance-Horan syndrome because she's going to pass that gene on to 50% of her children. Now, in males, NHS shows up as quite a severe cataract, much more severe than what she has. It has dental anomalies, and a lot of the boys will have intellectual disability as well. So this is a very useful thing for her to know as she grows up and starts thinking about her own family. And she can now take some actions to whatever she'd like to do, go ahead or not. So that gene has been around. We've known about that one for a long time. But this is a more recent gene that we've discovered. in the GenCat project. Now, this is another much bigger family, and you can see that all of the coloured ones there are squares, they're males, and the circles with a dot in them are the females that the gene is moving through. So those females with the dot are passing this gene on to their sons, and the sons are affected, but the mothers are not. So this is another similar situation where we have a difference between males and females in the rate of cataract. Just coincidentally, happen to find these ones. This is a different way of depicting the genome. This is a much bigger stretch of DNA. It's about 150,000 letters long. And the blue trace on the top is what it looks like in one of the unaffected boys. And so across this region, we're just looking at sort of the depth of information that we have, how many times we see a particular position in that genome. And the red trace below, hopefully you can see, has a big gap in it. It kind of just jumps a whole chunk of DNA. There's about 127,000 bases missing in those affected boys. And this deletion lines up with a gene there called PGRMC1, and it actually breaks the gene in half. So half of the gene is missing. So the cell has only got half the instructions it needs to make that product, and that's not useful. It disrupts its function significantly. So this was the first time that we had come across this gene potentially being involved in cataract. We didn't have any other families that looked like this. So we needed to do a bit more work to prove that PGRMC1 actually has a role in lens biology, to really nail down that we've got the right gene and can then use that in a future genetic test. So we developed up this zebrafish model. Zebrafish are a really great model to use in eye genetics research. They are small and easy to keep in the laboratory. They lay a lot of eggs, and they have a very big eye compared to the size of the fish, so it's easy to image the eye. So we take the single cell embryo within 30 minutes of the egg having been fertilised, and we inject it with what I'll call gene modifying reagents. There's a fair bit of molecular magic that happens in that injection to edit the gene that we're interested in. So in this case, we put in reagents that would knock out that PGRMC1 gene, so it wouldn't be available to the cell, similar to what we see in the family. And then we grow the fish up for 4 days and have a look at their lenses, and the unedited fish look like the bottom left there. The lenses are very clear, smooth structure, as it needs to be. And the one on the right is the edited fish, and the lens is taken on this really unstructured, I call it a sort of a scrambled egg appearance, and that is a cataract. So here we could show that if we knock out the gene, the animal with that knockout gets a cataract. So this is sufficient evidence that the PGRMC1 gene is now included in genetic tests for childhood cataract. There's a few more cases have been reported as well, which is good news, and other families can receive this genetic diagnosis also. So across the whole project over the last couple of decades, these are all the genes that we've found variants in our Australian families. And up on the height of the bar shows you how many families have a mutation in that gene. So NHS is fourth from the right. That's the first one I showed you. So that's actually the most common gene in our cohort, and PGRMC1 is somewhere in the middle. Oh, right next to NHS. There's only got one of those. So that just shows you there's actually quite a lot of diversity in the genetic causes. So across all of our families, we've only solved about half of the family so far, and that's considering all 280 genes that we know from right across all of the body of knowledge in science of what can cause a cataract. So we've got a fair bit of work to do to bring this up to where we'd like it to be able to provide a genetic diagnosis for any child anywhere who has a cataract. So our work is continuing to focus on more novel gene discovery, and these are two families we've been working with recently. Both of them have got gene variations in what we call a novel gene, haven't come up in cataract before, 2 different genes, one for each family, and those images are of the zebrafish where we've knocked the gene out. So this is great. We're very excited about this. But the variants in these families, we don't think they would completely obliterate the gene like we saw for PGRMC1. They make their small, subtle changes, and this model, while it tells us that the gene is involved in lens biology, it doesn't tell us what's going on with that variant. So we need to do something else to understand what's going on with that specific subtle change. So we're doing this in collaboration with a laboratory in Sydney led by Associate Professor Michael O'Connor. And what this group are able to do is take cells from blood, and we can reprogram them, turn them into lens cells. And then those little lens cells will aggregate together and form a tiny little micro lens. And it's only a few microns across, less than a millimetre. You can't really see it without the microscope, but it replicates the function of a full-sized lens. So if we then take these cells that can turn into lens cells, and we use the same molecular magic to edit their genomes and put in the variant that we're interested in, we can see if these little micro lenses will have a cataract or not. So that's where we're going with some recent funding that we've received from the NH and MRC to explore this work further. The other part of our current work is to understand more about the variants in the genes we already know about. So when someone has a genetic test, there are a number of results they could receive. Any variant that is found in a gene for the cataract is assessed against everything we know about that variant, how common it is in the population, how many times it's seen in people with a cataract, where it is in the protein, what it might do to change the gene's function. We gather all that evidence together and put it into one of these 5 categories. If it ends up at this end in the likely pathogenic or pathogenic zone, that's a positive genetic test, and that information can go back to the patient and be used to guide future decisions. If it ends up in the green end, where it's benign or likely benign, then we're pretty sure we haven't found the right gene and we need to keep looking or perhaps enroll them in our new gene discovery pipeline. But what we see is that about half of the variants in known cataract genes end up in this middle grey zone. We haven't got enough evidence either way. And this is a really frustrating place to be. It's probably that gene, but it might not be, because there's a lot of things that don't cause cataract in that spot as well. So to make progress in this space, we've picked a pair of genes. They're called gap junction genes, and they are one of the most common causes of childhood cataract when you consider both the genes together. They're related to each other and they do very similar things in the lens. The gap junctions are proteins. They sit on the edge of the cell and they join the cells together and make a conduit between the cells. So the lens cells will pass nutrients and metabolites and ions and all sorts of things through these gap junction channels. This is critical in the lens because there's no blood, there's no vasculature, there's no circulating blood. So this is the only way that lens cells can get rid of their waste or take in new nutrients. So about half of the variants we see in gap junction genes in our GenCat families fall into that variant of uncertain significance category. So we're starting with these genes to see if we can work out what's going on for these families. So as I said, the gap junctions form a channel between the cells for nutrients to move between. And we replicate that in the lab by growing a sheet of cells on a flat surface on a culture plate and then putting a scratch right down the middle of the plate. So we damage the cells in a defined location. And those damaged cells will take up the green dye. If the gap junctions are working correctly, that dye will then move out away from that scratch through the gap junctions, and that's what you can see in the left-hand picture there, where there's dye sort of spread away from the white line. The white line's where the scratch was. In the other image on the right, the dye has stayed only in those damaged cells along the scratch because those gap junctions aren't working properly. So if we then put the variant of interest into these cultured cells, and have a look at how far the dye moves from the scratch. We can estimate how well those gap junctions are working and use that to guide our classification of our variant, hopefully, well, probably up into the pathogenic side, but it could also move it down to the benign side and we can let it go. So those are the two main areas we're working on at the moment: the novel gene discovery, understanding the variants of uncertain significance, with the goal of solving all of the families in the study and making these comprehensive genetic tests that will help people all over the world. There's an army of people, obviously, who have been involved. And these are most of the current people, and thanks to our funders, the NHMRC, and to Cataract Kids Australia, who are a support and advocacy group, national for childhood cataract. Thank you very much.
Thank you, Kath. We'll bring you back up for questions afterwards. I'm sure Rufus always knew he had fish at IMAS, but he may not have known that he had fish at Menzies, so good to keep him on his toes. I'd now like to welcome our second speaker for the evening, Associate Professor Quan Hun. Quan is a part of our cardiovascular research flagship, and his work is looking at how to improve access to high-quality cardiovascular care in regional Tasmania. Thanks, Quan.
Thank you, Tracy, for the introduction. So hi, everyone. So my name is Quan. So my primary research interest in heart failure. So today I'm going to share some of our heart failure research findings and how we plan to bring heart failure care closer to home and make it more accessible for people living in rural and regional Tasmania and Australia. So what is heart failure? So heart failure is a syndrome that when the damage occurs to the heart muscle severe enough to prevent it from functioning properly as a pump. So sometimes that happens to the fueling function of the heart. So it can't fuel enough blood. But the other case, it happened to the pump itself, it doesn't function as a proper pump that can pump sufficient blood to the whole body. So the signs and symptoms of heart failure varies a lot. So the most common is, people will have short of breath, and other people may have, swollen feet, maybe cough, difficult sleeping. They also may have, impaired cognitive function and memory. And it's a clinical syndrome. So there isn't a single test to detect heart failure. So it depends on, the symptom, the size, and then the clinical assessment from the physicians. So in Australia, approximately around 500,000 people Australian living with heart failure, and about 40% of them are living in rural, regional, and remote areas. And people in rural and remote areas experience higher risk for death and rehospitalization compared to those living in metropolitan settings. And heart failure also the leading cause of also readmission among older adults, with around 30% at 30 days, and 40% at 90 days. So you can see that these heart failure patients, they come in and out of hospital frequently. Like 40% of them will come back to hospital within 90 days of being discharged from hospital. And these hospitalizations drive over $1 billion in annual healthcare costs in Australia. And one key factor that contributes to the high burden of heart failure is the multimorbidity. So what is multimorbidity? So typically defined as the coexistence of two or more chronic health conditions. The figure on the right is actually was originally designed for atrial fibrillation, but I see it's very relevant for heart failure as well, so I borrowed. So basically, multimorbidity can be classified into clinical, health behavior, and social and mental factors. And multimorbidity is highly common in people with heart failure. So in this study, we're looking at the roles of non-clinical and clinical factors and how they predict the risk of short-term readmission or death among heart failure patients. And beside the clinical factors like the severity of heart failure, you can see that, all the non-clinical factors like living alone, discharge during winter, or, remoteness index, indicate how remotely you live, also predict the risk of death and rehospitalization in heart failure. So we concluded that although the clinical factors are stronger predictors of adverse outcome heart failure, only when you combine the non-clinical factors and the clinical factors, that's when we can best predict the adverse outcome in heart failure population. So in this study, we specifically looking at cognitive impairment and depression and how they are associated with adverse outcome in heart failure. So for cognitive impairment, we use a MOCA score. So the MOCA score ranges from 0 to 30. So higher score means better cognitive function. So when you score between 26 to 30, that means you have a normal cognitive function. So that's why we see here that the MOCA score is protectively associated with outcomes among heart failure patients. For depression, depression symptoms, we use the PHQ-9, and for anxiety, we use the GAD-7 score. So for both the PHQ-9 and GAD-7, the higher score means higher depressive symptoms or higher anxiety. So here you can see that they are positively associated with the outcomes in the adverse outcome in heart failure. But overall, we can see that both cognitive impairment and depressive symptoms or anxiety, they are independently associated with the adverse outcome in heart failure. So in other words, people living with heart failure, if they have cognitive impairment and or depression or anxiety, they have higher risk of being readmitted very quickly to the hospital or die in the short term after being discharged from the hospital. So now we know the sort of factors that may predict the adverse outcome in heart failure. So the next item, we try to build a prediction scores that can predict the risk of people with heart failure. So we know who have more higher risk of being readmitted to hospital or die in the short term after they being discharged. So this risk score we use data from over 1,000 of heart failure patients that we recruit all over Australia. And you can see here that on the table on the right is including all the main factors that included in the risk score. And consistent with our previous findings that cognitive function and mental health are among the strongest predictors of the outcomes in heart failure. And also for living alone and discharge during winter. So now we have a tool to predict the risk of people living with heart failure. But there's still many unmet needs in heart failure care. So it's very difficult to manage heart failure, especially in the community. So, and because first reason that I mentioned before that it's because of the multimorbidity and patient complexity. So heart failure often coexists with multiple other conditions, for example, cognitive impairment, frailty, depression, anxiety. And there is an inadequate detection of fluid retention. So our reliance on the traditional auscultation for the breathwork, lung breathwork is insensitive. So this might lead to recurrent congestion or the fluid retention, which is a major reason for hospital readmission. And we have a very poor long-term adherence to treatment among people with heart failure. So as I mentioned before, the coexisting cognitive and functional limitation often impact the adherence to treatment and the lifestyle recommendation that we give to people living with heart failure. And lastly but not least, the limited sustainability of current disease management programs. So there are many available disease management programs that hospitals all over the world are applying to heart failure patients. But most of the programs are very expensive and it's not feasible to apply globally to all people with heart failure. And actually a lot of programs, they will abandon or even stop after a short time of implementation in real life because of the financial constraints. And our data show that there's a minimal benefit when we apply these programs to patients with low risk. So our proposal to overcome these challenges. So first, we developed a multidisciplinary disease management program that we have proven to reduce remission and mortality in heart failure. We also use AI-assisted handheld lung ultrasound. So this is a small device that we can plug into a smartphone or an iPad, and we do an ultrasound on the lung that we can detect the fluid retention. And this is much more sensitive than the traditional clinical assessment and the auscultation. We empower both patients and carrier through structured education and tailored cognitive intervention, try to overcome the long-term adherence. And our intervention apply a risk-guided approach. So we do a risk screening, identify the people with high risk who are more likely to benefit from the intervention. So to maximize the clinical benefit and the cost effectiveness of the intervention or the care delivery. So here are the components of the disease management program that we have developed. So as I mentioned, the handheld echo to optimize vascular volume, active charge, increase self-care education. So you can imagine that patients when they're in hospital, they are overwhelmed with everything. There may be delirium, so that it's very hard to digest all the information. So we provide the education after the discharge, send the nurse to home, repeat and reinforce the education that we provide during the hospital stay. We also provide exercise guidance that based on the patient functional status, so it's personalized to the individual patient. Close surveillance of symptoms, pulse, and other heart failure symptoms and signs. We also plan a drug titration and reviewing by cardiologists to make sure that the patient will reach the optimal dose of cardioprotective medications. We ensure a better transition of care through communication with the GPs, make sure that we have a care plan that well communicated with the GP. And as I said, we increase the intensity of health and nurse follow-up. So we do phone call and home visits after the patient are discharged from the hospital. So this figure was sort of findings from our clinical trial testing the effectiveness of the disease management program. So you can see that the x-axis is the risk score that we developed earlier. The y-axis shows the observed probability of readmission or death within 30 days. And the blue dots are for user care group, and the red triangles are for the intervention. So you can see here, among people with low risk, there was no difference between the two groups, user care and intervention. But as the risk increases, there was a split between the two groups, indicating that there are more benefits to people with high risk when they receive intervention. So this disease management program has been translated into practice, and the New South Wales government have reported a reduction of 180 hospitalizations per year in the Nepean and Blue Mountains, equating, of course, avoided around 2.5 million in that year. So then what about hospital care in regional, rural, and remote areas? We know that there's a fragmented and maybe a care provision inequities. So in our data, when we collect from 7 hospitals across 6 states of Australia, we found out that there is a regional variation in disease management program availability that largely contributes to the higher hospital admission rates in regional, rural, and remote areas. So our potential solution that we propose to deliver intervention remotely using telehealth, decentralized intervention tailored to the rural, regional, and remote population in order to ensure the equitable access to uniform high-quality health care. So that's when we developed this health outreach care model. Try to adapt and optimize our developed disease management program to improve health outcomes in rural, regional, and remote communities. So the care model will identify patients at the highest risk, integrate artificial intelligence in ultrasound analysis to detect, fluid retention, and deliver a tailored intervention remotely via telehealth. So we hope that we could address the disparity, the gaps in access to high quality public care. So this grant is still under embargo, so I shouldn't talk too much about the funder and too much about the grant details, but I can say that the study sponsor will be the University of Tasmania, and the study will start next year. And this is a collaboration between 2 leading research institutes, the Menzies and the Baker in Melbourne, 4 universities, 3 major regional hospitals, and 2 national GP networks. So this is the framework of the health care outreach model of care. So we have one central study hub that in Hobart that we will deliver all the intervention remotely from. And then we will recruit patients from 3 major regional hospitals, including one in the Royal Hobart Hospital and 2 more in the mainland, and also through the GP clinics all over across Australia. So basically, after we identify people with a confirmed heart failure diagnosis, we do a risk screening to identify those with high risk. So we choose so using the risk score that we developed, so we choose a cut point of 33%. So why 33%? So this figure you've seen before. So the 33% actually represent the median level of risk among a sample that we sample of half the patients that we recruited nationwide. And you can see that after the median level, the curve for the 2 groups seem to split, indicating that people with a risk over the median level tend to benefit more from the intervention compared to the usual care. So the eligible participant will be randomized to either usual care on the heart failure outreach model of care, and then we follow them up for 12 months and 24 months. So the significance of heart failure model of care is that we hope that this model will represent a transformative approach to heart failure management, addressing long-standing challenges in access, adherence, and early detection of the disease progression. And by combining the risk guided targeting, AI, lung ultrasound, and telehealth supported multidisciplinary care, we hope that this will extend evidence-based management beyond hospital wall and into the patient's home. And this innovation bridges the critical gaps in post-discharge care, particularly for rural and regional areas where readmission rates and inequities remain high. And the integration of the digital tools and personalized care pathway hopefully will ensure scalability, sustainability, and also real-world applicability. So we hope that this heart failure outreach will have the potential to redefine the national models, not only with the heart failure management, but also for other chronic disease management. So the next risk factor that I want to talk about is long COVID and COVID infection. This is a new risk factor, but probably relevant to most of us. Even though that the COVID, the acute pandemic already passed, people are still getting infected. And actually about 10% of all people with COVID infection have what we call long COVID. So they suffer from long-term, long-lasting symptoms. And that is the definition of long COVID. So you may wonder why long COVID. What does it have to do with heart disease? There is an emerging body of evidence that having long COVID or COVID infection actually increase the risk of your heart disease. So in this study, we compare between a cohort of people that we recruited before the pandemic and a cohort of people with long COVID. And here you can see that we look at the functional capacity, and we define impaired functional capacity by having less than 85% of predicted max. And we can see that people with long COVID, they're more likely to have impaired functional capacity compared to people without long COVID. And people with long COVID also have much higher risk of moderate and severe depression compared with people without long COVID. So in this study, we look at the association between long COVID and the heart function. So here, the association between long COVID and GOS. So GOS is the measure of cardiac muscle contraction. So we define that GOS, abnormal GOS, less than 60%, and reduced GOS at less than 80%. And you can see that people with long COVID are at higher risk of having a both abnormal and reduced heart function. So just give you a summary of our work in the long COVID. So since 2020, we have recruited around 1,900 people with long COVID. We link health data for over 5 million people who tested for COVID-19 in Victoria. And we also developed an intervention program for long COVID and tested it through an RCT called PERCEIVE. And the findings from the PERCEIVE trial show significant improvements in physical function in people suffering from long COVID. So our next step is to develop a perceived outreach model of care. So you can see there's similar to the heart failure outreach that we try to deliver intervention remotely to all Australian or any Australian who suffer from long COVID regardless of where they live. So there will be two study hubs, study central hub, one in Hobart and one in Melbourne. So this grant already came out embargo, so I can say that is funded by the Medical Research Future Fund. The study sponsor will be University of Tasmania. We haven't started the active recruitment just yet, but we are at the very last stage of the preparation. We're currently conducting the co-design with people with least experience long COVID, trying to make the study applicable to real work and fit their needs. So basically, what we do, we identify people with COVID infection. We screen them to make sure that they have long COVID. We screen the long COVID symptoms and to make sure if people have impaired functional capacity. If they do, they will be eligible for randomization to either perceived outreach model of care or usual care. And again, we will follow them up for 2 years. So if you or anyone, your family members or your friends suffering from long COVID, feel free to contact us, and then one of our team member will contact you or get back to you. Or even if you had a COVID infection before and you're unsure whether you have COVID, you can get in touch with us and we'll figure out whether you have long COVID and whether you're eligible for the study if you are interested. So here are some of my key take-home messages. So multi-mobility is the norm in heart failure. So the coexistence of multiple health conditions are very normal in heart failure, with cognitive impairment, frailty, depression, and emergent conditions like long COVID, jaundice, shaping the prognosis and the care complexity. The non-cardiac factors are powerful and under-recognized drivers of the adverse outcomes in heart failure. So the currently available heart failure care models are not really designed for multi-mobility, leading to suboptimal engagement and effectiveness, particularly for the people with living with cognitive impairment or frailty. And the next generation care should be personalized and multidimensional. So we should integrate cognitive, functional, psychosocial domains to improve the outcomes at scale. And finally, I think it's possible to bring heart failure care closer to home using innovative technology to ensure equitable access to care for people living in rural, regional, and remote areas. Thank you.
Thank you, Kwan. So our final speaker tonight, as we journey from eyes to hearts to now bones, is Associate Professor Dawn Aitken. And Dawn is going to tell us about her work on osteoarthritis and particularly the Impact TAS project, which is designed to increase access to physiotherapy and digital healthcare for Tasmanians living with osteoarthritis. Thanks, Dawn.
Thank you, Tracy. There we go. The slides are up. Well, thank you everyone for coming tonight, especially on a winter rainy night. It's very nice to see you all here in person, and welcome to those people online. Oh, sorry. There we go. Right, so I'd like to start by acknowledging the lived experience in the room. Tonight I'm going to be talking about arthritis and chronic pain. And unfortunately, everyone in this room will at some stage in their life experience chronic pain. And when I say chronic pain, what I mean is pain that lasts longer than 3 months. So pain that lasts longer than your normal body tissue healing process. There's going to be people in the room who are experiencing chronic pain right now, who have been experiencing it the last few months, years, and even decades. Tonight, I'm also going to challenge some of your beliefs and understandings about chronic pain. And I hope this is okay, because research is there to improve our understanding of diseases and to improve how we treat these diseases and ultimately prevent them. And the last thing I want to say before I start is that Australia has one of the best healthcare systems in the world. We're really fortunate to live where we are and receive the healthcare that we receive every day. But we can make improvements to this, and particularly how we treat chronic disease with our aging population is imperative that we continue to strengthen our healthcare system and the health of Australians. Tonight, I'm going to talk about high-value care. When I say high-value care, what I mean is that care that is supported by evidence of benefit to patients. It has a higher probability of benefit than harm and costs that provide greater benefits than other treatments. When I use the term low-value care, what I'm talking about is that treatments that do not have sufficient evidence of benefit to patients, or treatments that are recommended against in the guidelines, but we continue to use these treatments every day in clinical practice. Now I'm going to tell you a story about Mark. Mark was born in 1951. Growing up, he was a very fit, active, healthy young man. He loved sport like ice hockey and American football. And he loves the outdoors. Hiking, camping, fishing are Mark's passions. He's a really hard worker. He's worked construction his whole life. This involved really long days of manual labor, starting at 7 in the morning, often until 5 or 6 p.m. at night, so 12 to 13-hour days, 5 days a week. He worked outside. In winter, it was minus 30 degrees Celsius some days, a lot of days in winter in Canada, up to plus 30 in summer. Mark started developing pain from osteoarthritis in his joints in his late 30s. Like most people who develop chronic pain, they go and see their GP. His GP prescribed him some pain medication and told him that it was important to rest his joints when he wasn't at work. Mark really loved running, and he asked his GP, would it be okay if I kept running despite my pain in my hips and my knees? And the GP advised that it's probably best that he stop running and try and preserve his joints for as long as possible. Now, probably some people in the audience have realized that Mark is my dad. And today I'm going to tell you, I'm going to get a little bit emotional talking about Dad. I wasn't expecting that. I feel like I'm taking a, I know. So I'm going to tell you about Mark's osteoarthritis journey. Dad perceived for a long time with chronic pain, working really hard and also never complaining. You'd only know he was in pain because he'd be limping or he'd go and lie down at 6 p.m. Eventually the pain got too much and he had his hip replaced in 2005. And that was fairly successful. He was able to go back to work, but he still had chronic pain in his other hip and both of his knees. And it got so bad that he needed a hip replacement in his other hip in 2009. Things went a little bit downhill after this because when he tried to return to work, he just didn't have the function anymore. The 2 surgeries meant that he had a lot of muscle wastage. He had lost strength. He couldn't get down on his knees on the construction site and fiddle around with things or get down the ladders underneath the tunnels in the roadworks. And so he decided to take early retirement at 59. And then since then, he's been retired and he's had 2 knee replacements. And unfortunately, Dad is the statistic of the 1 in 5 people that remain in persistent pain following a joint replacement. So that's Dad today. He's 75, living in Canada. He's very stoic. He never complains still. And some would say he's even stubborn because he insists on mowing the lawn, even though he's using a walker now. He's quite disabled and can only walk 100 meters. So he mows a strip of his lawn, and then he goes and has a sit on his walker, and then he mows the next strip, And he does that for about 2 hours. And then he'll ask the neighbor if he wants him to mow his lawn. So that's Dad. And that's a really common story for people with osteoarthritis. So the term arthritis is an umbrella term for over 100 different medical conditions. Osteoarthritis makes up 90% of the type of arthritis that we see. And then there's other types of arthritis, which I'm sure you've heard about, that are inflammatory, like rheumatoid arthritis, scleroderma, psoriatic arthritis. But the main type of arthritis is osteoarthritis. Now, approximately 1 in 10 Australians and 1 in 5 over the age of 45 have osteoarthritis. And it mostly affects the knees, the hips, the hands, the spine, the feet, but essentially it can affect any joint in our body. It results in joint pain. That's people's main reason for presenting to medical care, because they're in pain. They lose function, flexibility, stiffness, tenderness, and crepitus. Crepitus is that noise of your cracking joints when you're moving them or going downstairs. Now, there's this widely held misbelief that osteoarthritis is a wear and tear disease and that it's simply a consequence of aging. We're living longer, therefore, it's becoming more common. But this is quite an outdated attitude that masks the reality that osteoarthritis is a complex condition. It can strike at any age, and it affects millions of people of working age. When you look at the risk factors for osteoarthritis, you can see how complex it is. Increasing age is only one of the risk factors. Occupations such as kneeling jobs and manual labor increase your risk. Mechanical factors such as malalignment or loading of those joints increase your risk. Muscle weakness increases your risk of developing pain in a joint. Family history and genetics. Female sex is a risk factor for developing osteoarthritis. We know it's more prevalent in females. But the two ones I want to focus on are overweight and obesity and joint injuries, which make up 70% of the risk of osteoarthritis. If we look at weight as a risk factor, we know that around 45 to 50% of the risk of osteoarthritis is attributable to excess body weight, which makes weight gain the largest modifiable risk factor we have for osteoarthritis. This is a pretty big public health opportunity because it's been modeled that if we reduced the body weight of the community by 5 kilograms, 25% of the people who develop osteoarthritis would not do so. It's also alarming that the AIHW released Australia's health statistics last month, and we have 67% of our population that's either overweight or obese. 27% of children now are overweight or obese. This is a major public health problem. So how do we diagnose osteoarthritis? Some of you might be surprised to know that it's actually a clinical diagnosis. You can have it diagnosed by your GP by just talking to them and them doing an exam on your knee. These are the things that your GP is looking for, and you don't have to tick all of them, but it's a combination of these things which would give you an osteoarthritis diagnosis. It's really common that people will go and have an X-ray, but we know now, after decades of research, that what we see on a scan doesn't correlate with the pain that people feel. So if we were just to do an X-ray on half a Launceston, we would find people who have osteoarthritis on their X-ray and have no pain, and vice versa. We would have people who have really bad knee pain, and we look and their knee might look like this one here, where they have nice joint space, there's no osteophytes. So the X-ray doesn't really tell us very much about the pain that people are experiencing. And another myth is that MRI tells us more. Okay, if an X-ray can't tell us what's going on in there, what about an MRI scan? Once you get over the age of 50, around 90% of people will have changes within their joints. So this doesn't relate to pain. It's sort of like changes we have in our skin as we age. We get wrinkles, but it doesn't mean our face is falling apart. That happens to all the tissues inside our body. And so scanning doesn't really tell you anything about what people are feeling and experiencing. How do we treat osteoarthritis? So our cornerstone first line treatments are education, exercise, and weight loss. This green bar at the bottom is what everybody should receive who has osteoarthritis. And then if they're getting this support and they're still in pain, they can move up to the next level around pharmacological pain relief. So they might look at things like Panadol osteo, maybe some anti-inflammatories to help dull down their pain as well. And then the top of the pyramid are the people that need surgery for their osteoarthritis. And if everything is managed well before this surgery triangle, very few would actually need their joints replaced. Now, this is a little small to see, but these are the recommendations of our key treatments. So exercise, education, and weight loss. And these are the guidelines across many countries around the world. And you can see a lot of green. So every guideline across the world recommends exercise, education, and weight loss. This is one of the pivotal studies that has really told us a lot about the importance of maintaining a healthy weight and your joint health. They did a very large randomized controlled trial in the United States. It was an 18-month diet intervention. Some people received the extensive diet intervention. Some people received diet and exercise, and then there was a control group. And what they found was the people who received the diet intervention, very intensive diet support. So we're talking, face-to-face visits with a dietitian on a regular basis, support with nutrition, education around what food to buy, all that sort of wraparound care. It resulted in 10% weight loss, around 10%. So most people in that group lost 10% of their weight, and they reported a 27% improvement in their pain. If you look at the group that had the diet and the exercise combined, they nearly had a 50% reduction of their knee pain. This has made its way into every clinical guideline around the world around the importance of weight loss. This was a really interesting study where they wanted to look at bariatric surgery and whether having bariatric surgery, so getting people to lose weight before you replace their joints, whether that had good benefits for post-surgical outcomes, so fewer revisions, fewer infections, fewer hospital days. When they did this study, the people in the lap band group got lap band surgery, and then there was a usual care control group. Following lap band surgery, 29% of the patients came off the surgical wait list and said they didn't need their joint replaced anymore, versus 5% in the usual care, which concluded that weight loss may eliminate the need for joint replacement surgery altogether in this group of people. Now, there's a lot of interest in the GLP-1 receptor agonists, including Ozempic. And I would say these have really great promise for osteoarthritis. But we do need to be careful because the weight loss that happens with these drugs is largely a loss of muscle mass, not fat mass. And that has detrimental effects on joint health. So alongside a weight loss drug, you still need to shift that behavior and try and change those lifestyle factors. This was another interesting study that wanted to do the same thing, look at mindfulness before people went to have joint replacement surgery. Similar to the bariatric surgery one, you're starting to see a trend here where people are researching how do we get better outcomes post-surgery, because that costs our health system a lot of money when people have surgeries that go bad and they end up in hospital for a long time and have many revision surgeries. Again, fascinating findings where people received an 8-week mindfulness training, and this improved their pain and function, and 30% of the patients chose not to progress onto surgery versus only 6% in the usual care group. So this is in some ways unsurprising because we know mindfulness is an effective treatment for chronic pain. But can we say that it prevents joint replacement surgery? And I'm going to talk a little bit about exercise in a few more slides, but just really highlighting here that exercise is still the most effective treatment that we have. and again, recommended in a lot of guidelines all across the world. Now, I've included a slide or two on injections because I get asked about injections all the time. And the University of Tasmania has just finished a stem cell trial. And I'll tell you the results hot off the press. But going and looking first at the guidelines, you can see a lot of red. So particularly focusing on PRP injections and stem cell injections, they are not currently recommended for osteoarthritis. But there's a lot of interest in potentially using these therapeutics. So we did a randomized control trial called the Sculptor trial with collaborators at the University of Sydney. And we randomized people to stem cell injections over a 2-year period versus a sham injection, which was just saline or salt water. So people had 50-50% chance of being randomized into your stem cell group or saltwater injections. Now, the company that co-funded this work, it was also funded by NHMRC, is called Sonata, and they're a stem cell company that's publicly owned. So when we analyzed the data for this trial, the database was locked. Statistician was put in a room for a week and crunched the data. And then we had to release the results to the ASX because it's a publicly owned company. What we found was that 50% of the active group and 48% of the control group improved over 2 years. So stem cells were equivalent to a saltwater injection in the knee. Now, Sonata has been putting a lot of R&D into this stem cell development for over 20 years. So this was a really heartbreaking finding for them. When this was released, this is the share market price of Sonata dropped down to 0. It was unfortunate that it's at the same time they were also developing stem cells for bone marrow transplants, and that trial findings also found them to be ineffective. So what I would like to say is that right now we do not offer stem cells or PRP injections outside registered clinical trials. They don't have disease-modifying or cartilage regeneration benefits. There's a strong publication bias, and the heavy marketing claims are not supported by the evidence that we see. Now, another thing that I hear a lot, and I'm sure many of you in the room have as well, is that osteoarthritis, it can only be fixed by surgery. The data really shows that around 1 in 5 people remain in persistent pain following a joint replacement. So it is not, it's not a new joint, is what I'm going to say. Now, we know it's highly effective for the right person at the right stage. The decision should be based on the patient's expectations. It's a patient-centered decision, but we know it's high cost. There is a failure rate, so 15% of hips and knees fail within 5 years. There's risks and also the dissatisfaction rates. And it's been modeled that we're currently spending $1.4 billion in knee replacements every year, but 200 million of this could be avoided with better lifestyle changes. So I guess it begs the question, why are we not using guideline recommended care for osteoarthritis? And there's a lot of reasons for this. We're still getting the word out there that exercise is safe, that running is good for your knees, that running is okay when you have osteoarthritis. You know, it takes a while for the research to get into the public knowledge. But what I'd also say is that our health system structure and funding is problematic in that we have primary care funded federally, hospital care funded by states. And this is a really nice quote that came out of last month from the AMA Tasmania when there was major ED block at LGH last month, where it says, patients do not care which government pays the bill, whether the bed is funded by the state or the Commonwealth, or whether the blockage is in the hospital, aged care, or the community. They just want the health system that's there when they need it. So the problem is that we really don't fund allied health well. It's easier for a GP to prescribe opioids for chronic pain than it is to refer them to physiotherapy. In Tasmania, we have some of the longest elective surgery wait times in the country and the highest rate of PBS opioids as well. So I'm just going to tell you very quickly about the GLAD program. This is an exercise program that was developed in Denmark, and it's for knee and hip osteoarthritis. You may have heard of it. We are trying to get the word out there about this program and how effective it is. And similar to our research around weight loss and mindfulness, it is a surgical avoidance model of care. So when this program is offered to people who qualify for surgery, 3 and 4 opt out of surgery 1 year later, and still 68% 2 years later say they don't need to have surgery. In Denmark, they have a Medicare code for the program now, something that we are working towards in Australia. It's in over 12 countries across the world, and in over 500 locations in Australia. Now, I'm conscious of time, so I just want to quickly say that we've implemented this program in partnership with the THS in Southern Tasmania. So we've implemented the program in Kingston, Clarence, and Glenorchy to look, is this program effective in a Tasmanian public context? And what we found was that it was effective. 85% of the patients reported clinically meaningful improvements in pain and function, and all 3 centers continue to offer this program now, free of cost. If you were to do the program in a private physiotherapy practice, where it is most commonly offered, it would cost you around $800. But we're offering it in public now for free. But it's not really for free because we're preventing joint replacement surgeries. This is a really nice quote from one of the patients. She lives out on a rural block, and she likes to go around with her dog, and she has hip osteoarthritis, and she was finding it really hard to do a bush wee. So she said, this sounds silly, but I can actually do bush wees, which before was just really, really difficult. And in the focus group, she was just beaming with herself. She was just so happy that she can move around her block and do the things she used to do. So I'd like to finish up by saying that this work was funded through the Royal Hobart Hospital Research Foundation initially, the Tasmanian Collaboration for Health Improvement, UTAS, Menzies, and a lot of the pilot work that we do. It's hard to get funding for this pilot work, so we really rely on philanthropic funding to support our research. And years of this philanthropic funding has led to us receiving a $5 million MRFF grant that's just started this month, and that will run for the next 5 years. We're expanding GLAD to 20 more locations free across Tasmania, and this is the lovely team that I get to work with every day to do this. Impact TAS is spreading the word about osteoarthritis and making high-value care accessible for everyone. So we're upscaling GLAD, and we're also upscaling virtual models of care. So we're going to be developing a Tasmanian-based MOOC, so a massive open online course for people with osteoarthritis that they can access in anywhere they are in Tassie. And we're also developing a virtual physiotherapy rehab program that people could have as well. That's Tasmanian-based. And so I did mention Tichy earlier as a funder of the Impact TAS work. And in my other hat, I'm the academic lead for the Tasmanian Collaboration for Health Improvement, which is a strategic partnership between UTAS, the Department of Health, Primary Health Tasmania, and Health Consumers Tasmania. And really what Tichy is there to do is improve the health of all Tasmanians by translating the research evidence and getting it into the practice. And we have received a grant last year that is around addressing high needs in Tasmania. This was a team effort, and there's 5 projects that are running to improve care in osteoarthritis, cardiovascular disease, high service utilization, Aboriginal health, and ED presentations. So it's a really, really exciting piece of work, and this is really demonstrating the value of this partnership. And if there is anyone in the room that wants to professionally connect and become a Teachy subscriber, there are flyers at the front. And last, I'm always looking for wonderful people to join our team. And particularly, we're looking for PhD students and honors students because we have these big grants coming on board now, and we need more workers. So yes, get in touch with me if you're interested in doing an honors or PhD, or you know anyone that is. And lastly, everyone in the room who has knee and hip osteoarthritis could join a study right now. We're recruiting that's looking at a lifestyle treatment for osteoarthritis. I can't tell you exactly what it is. You'll find out more once you look into it. But if you do join, it's a fully online study. So no matter where you live in Australia, you can take part, and you'd also get a free Fitbit. Flyers are up front for that one as well. Thank you, everyone, for your time.
Thank you, Dawn. Some excellent recruiting at the end, and that is true. We are always looking for great people to join us at Menzies. Can I please ask all of our presenters to come back to the stage, and we've got a little bit of time for some question and answer. So, can everyone still hear me? Terrific. Any questions for any of our speakers from the audience tonight? Right up the back, terrific.
I was just wondering, in the research into the children's cataracts, is it only passed down by the female?
No. I happened to choose two examples that were. That is coincidentally the genes that the GenCat project has happened upon. But no, most cataract is actually shared equally between males and females and can come through either parent. Great question.
Any other questions from the audience tonight? Another one here. Yeah.
I'd just like to support the last speaker. I spent 20 years thinking about getting my knees done, and 2 years ago I got them both done together. I went through the GLAD program. I was physically ready for the exercise, for the operation, but the results have been fantastic with no pain.
Wow. Well, I can imagine Dawn's going to maybe get your name and number, and you'll be up on a slide before you know it. Thanks so much for saying that and for endorsing that program of work. Any other questions? We'll just get a microphone to come to you.
Thank you. I also want to applaud the conservative approach to osteoarthritis management. As a GP, a lot of people come in wanting scans, surgical options, and there's a little bit of pushback, but I think the more we can get Tasmanians active in good physio and good health, hopefully we can prevent backlogs in the hospitals. And I just had a quick question about the childhood cataracts. Is the prevalence higher in Tasmania than other states?
No, it's about the same as everywhere. It's a fairly rare disease, but collectively across everything, it quickly adds up.
Yeah, thank you for your comment. And that's something that we really want to work with GPs around, because we know it's something where patients come in and they ask to have an X-ray, and so it's really hard when patients are asking something and you're trying to explain to them why they don't need that. And for a lot of people, the X-ray is like validating. It's, oh, I can see it. I have osteoarthritis. But I guess we also kind of need to always be thinking of the harms of the procedures and tests that we're doing. And there have been studies that have randomized people to being explained osteoarthritis with their X-ray versus just being explained clinically. And the people who are explained osteoarthritis with an X-ray end up believing that surgery is the only fix. They sort of devalue exercise in thinking, well, exercise wouldn't be able to fix that. And so there is some evidence generating now that doing X-rays is actually harmful care. And I think we have to be careful around how that's implemented, but I think it's really important.
Terrific. In the middle here, and then we'll come to the front.
How does the mindfulness work with osteoarthritis, the mindfulness? Is it visualizing what you want to happen, or how does this work?
Yeah, so I'm not a mindfulness expert, and I would say I probably struggle to sit still and do that. But it is a psychological technique where calming yourself down, and you're learning to be in the present mode, and being, feeling gratitude for the things that you do have. And we know with chronic pain that a large proportion of that pain isn't coming from the joint itself. It could be due to the stress that people are going through in their life, if they have low mood, if they have teenagers in the house who are all of a sudden gone off the rails. Oh, my knee has started to hurt now. And they have aging parents that they're looking after. And so the pain is actually so much more complex than we really understand. It comes a lot from here. So if we can treat our brain well, and by mindfulness is one way we can do that, we can start feeling less pain.
One last question in the middle there.
Thank you. I just wondered whether for young children, lens replacement is an option for improving their sight, if not dealing with all the other multi-mobilities.
Yes, that is. So cataract surgery in children, it's the same idea as in adults. You take out the damaged lens and put in an artificial lens. Don't always put it into very young children though, because their eye is still growing and the artificial lens can't grow with it. So it's often delayed for quite some time, and the children will have very thick glasses to compensate for not having an internal lens. But you have to do the surgery early, because if you don't take out that opacity, the brain, it sort of doesn't learn to see because it's not getting those light signals early enough. So it's a real balance. And I'm not a doctor, I'm a geneticist, so I can't give you any sort of medical advice or opinions, but it's a really tight balance about when you should do the surgery and then how to manage that child's vision so that their brain learns to see, and then can have a more appropriate lens when their eye is big enough.
Terrific. Please join me in thanking our three speakers. Take a seat. So, I'm really proud of all of our researchers at Menzies, but tonight we've tried to show three different examples of work that we do, but also there's a real story behind all of these studies. It's a story of starting with a small idea, scratching around trying to get it funded, putting together evidence, working with collaborators nationally and internationally, having the idea tested over and over again, and expanding that evidence that you may have to have it funded in schemes where nationally success rates in the NHMRC, the Medical Research Future Fund, sit at under 10%. So this is, you don't do medical research unless you are passionate, unless you're resilient. And I'm really proud of the work that not only you've seen tonight, but also that really characterises the work that's done at Menzies. So well done again, you guys. So before closing tonight, I do want to tell you a little bit more about how you can stay connected to Menzies and about some upcoming events. So there is many ways that you can help us on our mission: join a study, get involved through your lived experience, or support life-changing research by donating. And if you go to our website, you can find out about all of these things. I do want to, this is hot off the press and brand new just for the Launceston audience tonight. We are launching our consumer registry at Menzies. So this registry is about creating a strong, inclusive voice for all Tasmanians to be involved in medical research, so together we can improve health outcomes for our community. So consumer and community involvement means that people with lived experience, including consumers, carers, family members and friends, can help shape how our health research is planned, designed and delivered. And your experiences help make research more relevant, meaningful and connected to real life. This is another important way that we can ensure the community is truly at the heart of the work that we do at Menzies. So tonight we have Naomi with us. She's up the back in a green cardigan. If you want to learn more, she's our terrific Institute Consumer and Community Involvement Manager. You can have a chat to her on your way out, or you can grab some flyers from the table or jump on our website. Finally, I'd like to tell you, let you know that on the 9th of September, Menzies will be taking part in the inaugural National Medical Research Giving Day. So as a member of the Australian Association of Medical Research Institutes, we're joining more than 30 leading medical research institutes across Australia in a national giving day that celebrates the power of medical research to improve health, save lives, and create a healthier future for all Australians. So for nearly 40 years, our researchers at Menzies have been tackling some of our greatest health challenges, including dementia, multiple sclerosis, cardiovascular disease, cancer, diabetes, ageing and population health. So the National Medical Research Giving Day is an important opportunity to recognise the extraordinary impact of medical research and the essential role of donors and the community supporters in making that work possible. So on Giving Day, we invite you all down to Hobart. We'll be opening out the doors to our community, inviting them in to join us for lab tours and presentations, have a look in our clinics, and to celebrate as we come together to champion the research that changes lives.